We're going to kickstart this session. It's the roundup of the Roundtable Summaries. My name is Charlene Naik. I'm from the Walter Eliza Hall Institute in Melbourne. I'm also co-host of Ask the Doctor on Netflix. You should check it out. I'm also interested in hematopoiesis, lineage tracing, long walks on the beach, and chairing sessions to time. So, 12 minutes each, 3 minutes for questions. Let's go.
Sounds good. Hopefully I can be even shorter so we can have time for discussion. So I will summarize the ethics roundtable session that we had. So as everyone else, we had 3 sessions and quite lively discussions, I think, throughout each of our meetings. So one of the key discussions I think we had is how to balance access models in terms of the open to controlled access spectrum. So, we looked at risks, benefits, how do you explain these access models to donors, research participants. We also had several scenarios of what uses can be made of data, what are acceptable uses if you're looking at an open access scenarios, etc. For example, one case was raised, how do you manage access by or use of data by insurance companies, etc. So what are ethical acceptable uses? And another question was raised with respect to what is the enforcement in case of misuse of data. So if you're looking at typical controlled access models, how do you actually enforce misuse? Are agreements sufficient, etc. And as well, a middle ground approach was discussed in terms of registered access models. So it's something that's being piloted by the Global Alliance for Genomics and Health. And it's a bit of a lighter weight type of open versus controlled access, where you have some idea of who's using the data, how it's being used. A lot of discussion also surrounded identifiability, data sensitivity. So one of the key questions that's not always clear-cut is what is the threshold of identifiability for transcriptomic data or genomic data for that matter? I don't think there was necessarily a clear answer, but there was lively discussion as to how do you analyze this type of risk. There was also quite a few questions on how can you manage metadata, because as some of you may know, identifiability can also be seen as how associating certain types of data with the metadata that comes with it. So are there ways to coarsen metadata fields to make the whole data less sensitive, less potentially identifiable? So for example, there's ways of recording age, location data, etc., to reduce data sensitivity. And finally, some discussions were raised as to the use of data aggregation methods, adding noise to data, removing SNPs, etc., as potential ways of reducing data sensitivity. So these were discussed as, are these acceptable scientifically or not? So there's no clear-cut answer or solution, but they were discussed. And finally, there was also very lively discussion as to whether privacy is an illusion in this day and age, and can we expect privacy related to genomic information? Is this something that we should focus our attention on, or are we putting a lot of energy into it? So I think overall, this whole discussion on data identifiability sensitivity was one around how do we balance risks and scientific potential regarding the use of data and opening up access to data. Several questions were raised with respect to data withdrawal. So withdrawal is generally something that is explained to research participants in consent materials. So participants are offered with an option to withdraw data from certain databases. But of course, in an open access context, withdrawal becomes more difficult to implement. So the question of whether it is even possible to withdraw data sets from large databases is a good one, and how would this be implemented in projects such as the HTA. This is also related to questions of pseudonymization, for example, under data privacy regulations like the GDPR. How do you implement data withdrawal, and where would the codes be held? So those are questions I think that will have ongoing discussions within the community. An interesting point also is who can enact withdrawal of data for certain types of donors, for example, deceased donors, pediatric participants, fetal embryonic tissue or derived data from those tissues. Who is actually able to implement requests to withdraw data, if any? And this is in part related as well to how do you link these types of data? Who can give consent to data sharing of these types of tissues, etc. There was also a question in this matter about whether if you're providing data about one of these vulnerable populations, you're also providing data about family members, relatives. So the question of identifiability becomes a bit wider than just data about that particular individual. And finally, there were a few questions about trust building within the HCA, within the scientific community more largely. In particular, the role of the scientific community in building trust with participants, with society, was something that was raised. Some discussion suggested speaking to politicians regarding use of data, enacting legislation to protect, discrimination based on genetic characteristics. Is this something that the scientific community should be lobbying for to ensure that we can broaden data sharing while having some kind of societal agreement on limiting uses? And as well, there were comments as to how do we appropriately inform donors, participants of the risks relating to use of data, especially in open access contexts where future risks might be unknown. And so how do you formulate this kind of unknown future risk in this community and be respectful of wishes of the donors once they consent to data sharing? On that note, I'll welcome any questions, and if I've forgotten some of the discussions, please feel free to add points. Thanks.
Questions from the audience? Yes.
Not a question, but additional comment. So if in Europe, so there is a data protection law, but there is an exception law, the exception law 28. Then, so if the data is anonymous, then also even so the patient withdraw the consent. So we don't have to delete the data. Also published data, also if it's anonymous, then also the exception law number 28 applies. Then also there is a kind of sentence in the law that's also even the patient withdraw the consent, but still we can still share or we can still publish the data. So if it's technically impossible, so that means that this European law also accepts some kind of exceptions. So that's what I wanted to comment on that.
Yeah. So indeed, under the GDPR, I think withdrawal for data sharing purpose is a complex issue. It's something that's an ongoing discussion with the GDPR task force, with also the DCP in terms of how this would be implemented. It remains that data withdrawal is still an ethical concept that many ethics committee expect to be addressed at least. So there are mechanisms of, how would you handle such requests? How do you explain, possibilities that data cannot be withdrawn to participants? So it is complex under the GDPR, but I think it's also a wider issue in terms of research ethics and how do we think about this and how do we explain it to donors.
So I'm at a children's hospital, and we just published a data set on pathogenic effector T cells in eosinophilic esophagitis. We have treatment-resistant and treatment-sensitive individuals, and not a huge data set, but what we did was take the SNPs out and just uploaded the reference mapped reads. So we still had read files up there for people, but we don't see a way to consent for kids, period. We just don't see it. So, but we have, you want to share sequence level data, and it's so that it allows for, more creative analyses. What you said there was a big argument about removing SNPs that it wasn't attractive. What's the argument against it? I think that we have to do that as a step one for open data.
So I'm not a technical expert. I think there are people in the room who are better placed to answer this question. There.
You know how they can tell if someone smoked a cigarette and what color the hair on the subway platform? My argument is sometimes you could have a rare variant that you have a particular expression signature for, so even if you remove the variants in the expression data, you might be able to trace that back with enough metadata plus that genomic signature for that rare variant. That's all I was thinking. So I didn't say it was not attractive, but I'm saying someone will probably come up and say a paper and say, you guys are doing it all wrong.
I know some people in the audience who'd be scientifically upset about SNP removal, and does anyone want to comment on that? And also the chance of detecting it at 3' versus full-length reads? That can be a second question. Maybe you've just got first comment first.
I guess mine's a little tangential, but just wondering about both the private and the public what am I lost my thought? The personalized medicine initiatives. Mm-hmm. And I mean, the connection between those and the single cell data from the HCA could be enormously powerful for sort of medical advancement as you sort of link those genomic SNPs to the expression data. And I just wonder how it's sort of tangential to this question of ethics within this system, but what people think about that.
So the question of data linkage is quite a complex one from an ethics perspective and from a scientific one. So, of course, there are advantages to be able to link data sets, scientific advantages. The question, I think, is you need to probably be able to explain this to donors ahead of time. That would be my inkling. Or you'd be able to, be very careful about your consent wording and how you word potential for re-identifiability, because linkage is in a way linked to putting data sets together. You're increasing the sensitivity. You might be getting additional information that the original consent did not cover for one project, for the other project. So I think, all in all, we have to be careful about how we think about linkage, how we present it, how we explain it. We explain that one database may have your information, another may have as well. And if we put it together scientifically, it's interesting, but there are privacy risks associated to it. So it's finding the right wording. I think explaining it in the clearest way possible is one of the objectives we can, perhaps as an ethics committee, work towards.
Any other questions or comments? Righto. No, there wasn't. I was brushing your hair back. Okay. We might move on then. Thanks very much.
Thank you.
So the next roundtable we'll hear from was funding and cross-collaboration with Jonah Cool and Katrina Gold, and looks like Jonah will give a summary. Thanks, Jonah.
Yeah. Thank you very much. My name is Jonah Cool from the Chan Zuckerberg Initiative, and Katrina Gold from the Wellcome Trust and I sorry, I'm just getting my laptop set up here, host a discussion on collaboration. I don't know if we have another slide. We did not make slides. Collaboration and communication among different consortia. And this was a very nice discussion. I want to thank the many participants that came and included both some of the funders that are here and represented from around the world, as well as scientists from many different groups and countries. And I think at a high level, there were 4 emergent themes that came out of this. One was still some understanding of who exactly is a part of the Human Cell Atlas. And this is true both at the level of different groups that are funded and different projects that are participating and contributing, but then also as a whole. And in particular, I think a real curiosity among the community of not only who those individuals are, but who has common interests, who are the people close by that they might get in touch with or learn more about the HCA. And so there were a lot of kind of threads around contacts. So I think many were looking for a national level contact. So in Japan, for example, who exactly can they reach out to get more information, as well as more thematic. So on a particular topic of a tissue or computational method, I think clarifying some of these, both at the level of projects that are funded, but then also at the level of the larger community, is an area where I think all of us can improve, and we learned a lot. There was also some questions around whether funding is a contingency of participation. And again, I think both as funders and as a community, we can hopefully work to clarify this. And I think this is also a good opportunity to get maybe some questions from the live stream, to hear from those that are both here and then also remote. And so then the second level or kind of high level theme was around updates. And I think a lot of this stemmed from the enthusiasm on the updates earlier in the day, and the fact that people learn a lot from these very high-level views of what's going on and who's involved and who's funding these projects, and how to do this on a more regular basis. I think that we were kind of excited and maybe slightly surprised that there was a real appetite for regular updates from, again, individual projects that are being funded, but then also maybe at the consortia, potentially at the weekly, bi-weekly, or monthly basis. So, when we asked, or the discussion got to the point of, what level or how frequent these updates would be, it was pretty regular. And so I think there's a real hunger for this sort of information, and I think it's worth again thinking as a group of how we can do this better. And then I think one other thing that came up, and this maybe relates to this clarification on the updates, is a sense at times that certainly when projects are funded, or maybe as the community makes progress, that many of these topics are, come off as being polished and done. And for new members of the HCA that are looking to get involved, it can be very difficult to quickly identify what is to be done and where their specific contribution can be. And I think that this is a really, really critical item, given that there is so much to be done, and it's not at all to belittle or understate the amount of progress that's being made, which is really impressive. But there's a lot of work, and I think that everyone would be excited about having more contributions. So trying to make sure that we highlight those areas and make it as easy as possible for new groups to become involved. There was some discussion on once maybe a new member becomes interested and involved, how they might get to more granular level of information. So there was a lot of discussion around Slack and email and the newsletters. I think we actually often came back to some ideas around message boards that might allow people that aren't part of individual projects to kind of understand what was going on, to either look for a common thread or potentially ask for help or advice. And then the final topic and kind of high-level item that came up was around policies, and a request and a recognition that within the HCA community, it's being supported by many funders. And while that's good, it also at times might mean that there's slightly different policies that each funder has. And to the extent that we can, again, as a community, clarify what the policies are for the Human Cell Atlas, and then as funders, try to support those as best as possible. And I think that this has been something that we've tried to do. So I think both the Wellcome Trust and CCI, as well as many other funders, have tried to explicitly support the community policies getting better, both as funders to support them, but then also as a community to really explicitly clarify what this means in terms of data sharing and what it means in terms of many other aspects of the ethics and other large issues. So with that, I'd be happy to take some questions, or would also encourage others that participated to contribute.
Thanks, Jonah. Plenty of time for questions, comments.
One of the things we talked about, and I think I was lucky enough to be in the first session, is not the requirement, but the what's the word I'm looking for? The need for people who are in the PCA to have some responsibility to the HCA, sorry, to have some responsibility to the HCA. As in, you know, if you're a member of the HCA, you should be doing something for the HCA in some way, even if you're just a member. And one of the things maybe we could do is have that reporting or that update be one of our requirements as part of the HCA. You know, in other words, just give an update to the HCA project about what you're working on relative to the HCA, and that might satisfy it. I don't know what other people think, but that would help. That would go a ways towards letting other people know what other people are working on, and usually when I'm part of a certain consortium or center, I have some requirements that I have to fulfill once a year, teaching or whatever. But and I'm sure the HCA doesn't want me to get up and teach a class, but maybe just report what I'm working on.
Yep, thank you.
So does anyone who's in charge of that or thinking about the HCA newsletter want to comment on what that looks like and how we might better facilitate that?
First on, I think Deanne was asking, right? Yeah. On people updating on their own activities, that's the idea of the project registry, and we can choose as a community to be more prescriptive and demand or leave it to people's own best judgment. I think the more all of us model real information there, the better off we would be. And one of our goals for the next few months is really to improve kind of our tracking system so that that information will be available and conveyed. But it really behooves all of us. We're not gonna, I think policing is just not our culture. But I think we can model great behavior around that, and many of us can provide more information there than we have before. And some of it is just, it's time consuming to provide information. And so there's always something better to do at any given moment. So maybe I think the interface could ask us for more information and then could make it easy to provide it. There's also, I think, the hope that once the DCP is up and running and has data releases, a lot of these issues will go away because you could just go there and see what's there. So for anything people have already uploaded, you would have this information. It wouldn't tell you a lot about what people are planning to do. So these are two different aspects. In the newsletter, I think so far we've focused more on activity updates than on content updates, meaning on what's in the HCA and what people are doing, precisely for these reasons. Once a project registry goes beyond, these are the systems people study, down to the level of, and these are the experiments they're actually doing, I think the registry would provide information that we can also summarize into the newsletter. Thank you. And I think in this context, really a big shout out to Jenny Rood and to Tracy and to Jane for putting the newsletter together, because that's actually a volunteer activity.
Could I get a show of hands related to Jonah's point 3? For those who are not yet officially contributing to the Human Sellout list, could you put your hand up? But would like to? Yep. Now, could you put your hand up if you don't actually know what the Human Sellout list really is? Everyone knows what it really is? I don't know what it really is. I guess the point I wanted to make is that it's still evolving. I mean, it's not like there's every mechanism in place and every tissue has a lead and there's a very clear direction. It's something that is really a consequence of self-assembly, and we were talking about that in the OC committee. So there was a sense that I got from people over tea break that, like you were saying, that it feels like everything's already done, or how do I get involved? So I think the answer to that is that it's not fully known what needs to be done, but there's an openness to bring people in. But what the mechanism to do that is something that's not yet established, but could be worked on.
And I think some of these, early efforts like the newsletter and I think continuing to evolve the registry and, even meetings like this are really helpful, right? I think that they are wonderful opportunities for people to get to know those that are deeply involved and those that are new. And so, figuring out how to scale this up, and it's the challenge.
I just wanted to add, Chloe made a point in the final session about how HCA is a community rather than perhaps a formalized consortium. It contains all kinds of consortia and groups of people. And I think from the start there's been this idea of real openness and inclusivity, which is wonderful. But I wonder if sometimes it actually presents a barrier because people think somehow there must be more, more rules than there actually are, and perhaps a little bit more definition around those structures might help even further outreach.
Yep, down the front here.
Jonah, I was just wondering, was there any discussion about leveraging funding? So with the philanthropics, there's certain amounts of money available. Are there conditions built in that the applicants need to try to leverage funding from their national governments, from other agencies?
So I don't know for all funders. I can speak for ourselves, and I think probably true of Welcome as well, that's not a condition. That being said, I'm sure that part of getting involved is there is a desire to grow these projects, and that might include additional funding from the initial source and also additional sources. And there were some wonderful examples of that earlier today. I think that this also maybe relates to this point of trying to be as open as possible of what is left to be done, right? So that the funders, and I think that this, among the funders group that participated in some of these roundtable discussions, identified that, to the extent that it's easier for them to understand what other international funders are doing. And so, this isn't just about transparency and communication among the scientific community, but, the entire community here. So both at the level of funders and also the scientists.
And also for the cross-collaboration, was there any discussion about building in conditions about international components that there needed to be applicants from multiple countries, for example?
So yeah, there were a couple, thank you for raising this. And I think maybe the first discussion, this related to the policies. So again, as a community, if there were policies or encouragement on, geographic diversity, gender diversity, ethnic diversity, you can imagine quite a few points here that are really important. So I think, again, encouraging and trying to identify exactly what as a community is the right thing to do there, I think is something that people are thinking about.
Any other questions? Comment?
I'm slightly tangential. I really appreciate what you guys are doing, and even though I haven't gotten 1 cent or 1 nibble on the grants that I've submitted, I really appreciate what CZI is doing to stimulate this community. I'm wondering about, when you think about what CZI is trying to do long term, and what you really want to accelerate to. And then you look at what's going on in the HCA space, and where there's super progress and where there's maybe less acceleration towards those end goals. What are you thinking about bridging to those next goals, and what sort of efforts are going to take HCA up to that higher impact level? Where it's really translating, and not just like in niche places, but really transforming the way we think about biomedical research and the future of research, and then improving human health and all that stuff. So I know that's really the core mission, and I believe in it greatly. So yes. What is that? Given that, how does that change our agenda, and does that give us some new agenda points in HCA to?
So I'm going to deflect the question a little bit, and I'm happy to talk at the break. And part of this is I don't want to, speak just on behalf of CZI, because quite honestly, I think that the reason that CZI is excited about this community is shared among many funders, and that is that there is a belief and a recognition that there's a huge amount of progress being made here that is applicable to a large number of tissues, right? And there are some funders that have a very specific interest in a given tissue, so Garabet and the Helmsley Trust, but then also many others, the Japanese funders that are here, the Wellcome Trust, CZI, that recognize that both the power of the community and I think the way that they're approaching the science, but then also this technology is critical. And part of roundtables like this are trying to understand what the needs are for the community to move forward and supporting those. So I don't know, so that clearly did not answer your question in specific terms, but I think that suffice it to say that we've tried to be very supportive up until this point and will continue to be, and I think there's many others that feel similarly. And I think it's the trajectory and the arc of the community that we're very excited about.
All right, we might leave it there. Thanks very much, Jonah. So next up, it's how to engage with the DCP. That was chaired by Gabby Yordanova and Mallory Freeberg.
Hey everybody, I'm Tim Tickle. Also Mallory and Gabby are here today to talk a little bit about our DCP engagement table. And I'm very excited to have the kinds of conversations with everyone. We had people who were interested in technology, they were interested in engaging people, and they were also interested in engaging our developers. In fact, we had someone sort of break off on the side and work with one of our developers on playing with some of the APIs, which was really cool. So the kinds of main takeaways that we had, number one, people were really interested in just how to work with the DCP infrastructure itself and the system. People represented tools like methodology, for instance, portals, databases. And so we talked a little bit about, number one, that we have a portal community where people come together and talk about how to engage DCP as it's maturing. We have some initial examples of that, but we're still growing that space, but we're learning how to do that. And also sort of how would one integrate, for instance, or suggest a tool, for instance, for a pipeline for it. And so we got some feedback on how that might be the case, and we look forward to working with community members, getting feedback on the kinds of tools they might be interested in suggesting to the pipelines. Other thing as well that we mentioned was that we have a methods registry and a portals registry in our website, and so that is something that you can use to get the word out on some tools or portals that you have that you'd love this community to be thinking about. As far as pipelines goes, pipelines were also something that people were really interested in, and we're excited that you are. There were some questions about the benchmarking and how we do it. We have reports that we work with our analysis working group on as far as it's our process of making these reports so we can see that pipelines make sense to use. And so we'll make these, they're publicly available, but we'll make sure that they're in a space that's more present for people so that they can see these benchmarking as well as we're working with our AWG members. Also, there was just a question about, in general, how does one find documentation on the outputs? And obviously, the outputs, we try to hit them well and have good outputs for you, but they're also a thing that we can improve and iterate with you. So we want to make sure that you can see this documentation so that you give us feedback and suggestion. And so although it is available on our website, once again, we're always interested in hearing about what other kinds of feedback you'd be interested in. As far as submitting data, we had lots of questions about how to submit data and our metadata model, the ingest process, and just access control, how will that ever happen with our system itself? And so we do have some documentation for the metadata model, and we do have individuals that would work with you on the ingestion process. So feel free to reach out as far as that goes to make sure that we have you in mind as you're thinking through submitting to the DCP. As far as access control goes, that's something, of course, has been communicated to us, and we're thinking hard about how we pull that into our roadmap. In general, there was a lot of questions about how to get more specific communication. And so, for instance, a lot of exciting people making really cool portal solutions. And so we mentioned the portal community in our Slack channel. There's a link here for the Slack channel as well in case you're interested in joining our Slack, HCA's Slack space. We're hoping that will be a space where you can either join the conversation or be a part of the meetings. As far as visibility of software documentation, the updates for the document, our pipelines themselves and other components are available in releases, software releases as we go. But we're interested in hearing more about what kind of way you would like to engage that, because we understand that might be something that is a little difficult to approach. So that in general, we had questions about software documentation and DCP. So, And then lastly, there were a lot of questions actually on what our approach was to APIs and what APIs were available for retrieving data. And we do have APIs available. We have Swagger for them for our developers. But if there are certain kinds of APIs you're interested in, we'd love to hear even more. Currently, we do have some APIs for data store access, and we have some APIs to access our matrix service as well. And we're building more for query and others. So those were the general takeaways. Anything else to add? Cool. And so maybe we did this short and sweet, but I'm open to questions if you have any.
Great, thank you. Questions and comments from the audience? At the front here, Jay.
Great, thank you. And I guess related to the previous point of how to engage with the human cell atlas community is also depositing their own data and being able to browse their own data and integrate their data with other people. So I guess it's a question that everyone will be asking is when will it be fully open for full use access, and how can we start depositing our own data for comparative analysis and so on?
Well, as far as depositing data, you can do that now. We're very interested in working with you. So if anyone here is interested in working on depositing data, please just contact us and let us know.
So it's through the DCP team for depositing?
Yeah, definitely.
Will it continue like that, or will there be kind of an open ingesting system that will allow people to upload at will?
Yep, go for that.
Yep. So we have like a rolling submission right now. So really, I'm one of the data wranglers. There are 6 of us. So we interact closely with contributors who say they have data that might be ready to contribute. So we work one-on-one with them. But yeah, it's a continuous rolling process. We may in the future have maybe calls for data. I'm imagining maybe 10 years from now we realize there's not a lot of skin data, so maybe we'll do a call for skin data. But right now it's just on a rolling basis.
The back.
Hi, this is Neil Skellenberg from Harvard Medical School. One question that I'm very interested in is, and I didn't bring it up when I was at your table, is about file formats and structuring data. Since you're talking about integration with other portals, you have to come up with some sort of a format in which data will be exchanged. But of course, the same is true for any package that someone might write in R or Python. What is your process for that? And is there a way for collaboration across the other consortia and others in the community who have maybe already built similar things or agreed on or come up with formats?
So our general strategy is, one, if there's a GA4GH standard, we adopt it. If there's not, then we work with the community on what makes sense as a de facto standard until something is established in GA4GH. So like, for instance, we use SAMspec. We don't use SAM files, but we use SAMspec. We use BAMs, for instance, in the actual system itself, because it's a GA4GH standard. But for expression matrices, there's not really a standard for that. And so we looked into the community upon what kinds of file formats, schema, or containers that would be interesting for them. And we trying to make sure we're offering those until there's more guidance from GA4GH. Does that answer the question?
Partially.
Yep.
So in the case of those situations where there is no guidance from no existing guidance, do you go through some community process, or do you have a set of friends who you reach out to, or how do you do this? That's a question that I'm thinking about in the context of other projects like HubMap, for example. Do you have any recommendations?
So we have a couple ways of engaging the community. Firstly, we have an analysis community that we meet every 2 weeks, and we certainly get feedback from people there. As well, we have a very active team that tests the system and gets feedback from our users. And so we are also be able to get feedback in that way as well. And in an agile way, develop and make sure that we're hitting what our users need. So both beta testing and also engagement of the community.
Yeah, at the back.
Sorry, and just to add to Tim's answer, so I'm also a member of the DCP. We are also proactively engaging with consortia like HubMap and HTAN to try and align on standards. That's very early in the process. There isn't any alignment yet, but we are actively pursuing that.
There's a question down the front here.
Oh, I was just going to say one last thing about data formats. In cases where we don't have a single opinion about the data format, we're actually trying really hard to build it into the system, an ability to receive feedback from our users. So for the Matrix service, for example, we provide a couple options for our users to consume. We hope that one of those options will fulfill their use case, and eventually that we're able to learn from that and understand what the community needs in terms of the formats. It's a lot better for us to be able to build it from the bottom up whenever possible.
That's great, thank you. I just have maybe one more point to make, since I think with file format, something that is so painful in bioinformatics that we all hate. You know, and when you put something out in a certain format and other people start building their tools around that, and then you decide, well, maybe this was not the way how we should have done it. I'm wondering if there could also be some sort of, you know, built-in versioning from the start, so we all know what we're dealing with.
Versioning for the DCP in general, the concept is definitely for lots of different things is in the system or being put into the system. Yeah, I agree with you. Yeah.
Any more comments, questions? No, all right, we might leave it there and get on the next group. Thanks very much. So the next roundtable was Querying the Atlas, led by Dana, John, Peter, and Irene. And Dana will summarize their findings.
Okay, so we basically tried to engage in 3 questions. What is your biological question? What method do you need to answer it? And what would you like to visualize this and anything else? We talked a little bit about visualization, but mostly about some basic missing things. And there were 3 different groups, but there were common themes. The first one is pretty much, and actually I find it funny because I actually had 2 queries or general use cases that were brought up in the computational meeting in Stockholm about 2 years ago, and they were the most discussed methods. Pretty much, how do you use the atlas to compare normal to disease? So the atlas will have a lot of great healthy data, but now I have a disease sample, and there was a lot of discussion how this problem is still complex, and still today there's no tool out there that does this well. You have to really distinguish how do you match cell types between healthy and disease, because they've changed a lot. Is it a new cell type? Is it one cell type to another? How do you quantify composition of cells? How do you, again, account for things? How does the tissue architecture differ? And really the main stumbling blocks is what we talked about today, data normalization. In order to get a good answer for this, first of all, you need data normalization. We haven't really, as a community, defined what a cell type is, and for most of the methods that our community is trying to develop, a lack of ground truth is really lacking. So while this is a query which everyone wants, it's one of the ones with the fewest sort of tools that people are happy with. We did manage to, really make some progress in trying to figuring out how do we address this. So comparing between samples is a general problem. Also longitudinal samples, there lacks really good tools for longitudinal samples. And we thought, well, maybe as a community, we should collect some ground truth data and make it available for the computational community. Of course, we want to look at it at multiple modalities. The more modalities, the more cross-referencing you have to get some idea that what you're finding makes sense. So the RNA, the epigenetics, and the spatial data. But really, if we can take, for example, a mouse model of disease such as cancer, which a lot of people are trying to compare cancer to normal, and look at the early progression of cancer. So you get longitudinal data, smaller time steps as the disease progresses, that will allow you to connect the dots, generate some ground truth that will help you collect the 2 extreme endpoints. And this could really help develop good methods that can be used in the human case where you don't have this slow progression. So we think that this could be a very valuable training data set for the community. The second most frequently asked question was things to how can we use this data to learn or infer regulatory networks, predict response to some type of perturbation, whether it's CRISPR or a drug, predict how to push or to convince one cell type to reprogram into another. And we had a few discussions about this. The power of statistical dependencies in single-cell data, which really is an enormous power to actually infer regulatory networks. But then you have causality versus correlation. We think that now with the single-cell ATAC-seq methods coming on board, integration between these can really open up a lot of opportunities in asking regulatory questions. One idea was we could replace ENCODE, and by combining a single-cell RNA-seq and single-cell ATAC-seq, we can really get a fine map across many, many cell types for TF to targets, again, computationally inferred. And again, like every other computational problem, you know, as you're developing these things, does it work? Does it not work? There's real lack of ground truth. Here, the idea of a good ground truth data set could be going from organoids to CRISPR perturbation. And the third is the simple scenario of blasting cells. And here we discussed that, again, we need normalization. Normalization comes up again and again and again when you want to compare data sets. And the fact that we haven't quite yet defined what is a cell type, what is a good similarity matrix, we figured out that not all genes are created equally. So you might want to do some feature selection and figure out when you want to blast a cell, what features should you use? What similarity should you use? How should you weigh the different features? And again, a lack of ground truth came up. And here, the annotation platform that Peter talked about earlier today, we thought could be a very powerful thing in providing a ground truth, both for defining what is a cell type, what is a good similarity matrix, and do you have good blasting algorithms from one cell type to the next? So again, an effort that's already undergoing will provide the ground truth training to address this use case. And finally, a visualization request is, as we've been really focused on cells, a lot of our visualization is cell-based. There's some gene matrix visualization, but now with the rise of some of the epigenetics data, the single-cell ATAC methods, we need to begin thinking about how to integrate genome browser-like views that look at pileups, look along the genome, and can look at both the single-cell ATAC data and possibly chip data in the future, should it be single-cell, the actual gene expression data, again, in the context of the chromosome, and also integrate and look at how this compares to some of the legacy data from places like ENCODE and other consortiums that have collected such data at scale in bulk sorted sets. So those were the main things we talked about. And generally, we think that use cases are still missing. So again, this is a call to the biological community. Each one of these tools that need to be developed, what is a good similarity metric? What are the meaningful differences between disease and normal? Everything is a trade-off. Every normalization is a trade-off. And really, the way these methods are going to be good is if computational biologists and the real experts, domain experts, pair up so that we can get the most important features to conserve in our normalization, the most meaningful answers from comparisons, the most meaningful blast, and what really gives the biology and highlights the real biology that you need to get. Because, there's statistics, there's mathematics, you can build different similarity metrics, there's different statistical ways of saying this matches this, but not all statistics highlights the same quality of biology, and this is why this effort really needs to be a closely joined effort between domain experts and the computational scientists. And to give the biologists the tools that they want, they have to engage and define what they need. And that's it.
Thanks, Dana.
My other co-chairs want to join and answer the questions? No questions.
There must be some. Well, maybe I'll start off first. Someone in the audience raised earlier today how non-experts might be able to interrogate the atlas and are some of these questions you raised. Was that a topic of discussion? Or do you have any thoughts on that if not?
So again, some of these use cases, once there's tools that are robust and reliable, are exactly the type of tools that you'd want to make available. For example, this BLAST tool that we were describing, I can imagine, half the biologists, right now BLAST is something you learn in an undergrad year 1 biology. So again, if we have a good BLAST method, if we have a good similarity metric, once Peter sets up the annotation database, then yes, we can make a BLAST that will be as simple to use and as robust to use to the wider community. Again, to get these things robust, they have to be done right. To be done right, the needs of the biological community have to be defined. And we're an emerging community. There are a lot of tools out there. There are a lot of visualizations. There are a lot of computational tools that have some pretty easy to use interface. What some of these tools don't have is guardrails to make sure that you avoid falling into artifacts and pitfalls, and make sure that the result that you're getting from these tools is a reliable, robust, sound result. So I think it's our responsibility as a computational community to provide as many tools as possible with guidelines on how to use them and guardrails so that they'll be, they're pretty robust to giving sound scientific answers, even with a little bit of misuse. And it's the biologist's responsibility to really tell us what matters, what questions they need answer, what they prioritize, what signal is meaningful for them, and what's most important for them to get out of it, because the challenges are endless. If we're to build a roadmap, as Aviv suggested, we'd love to prioritize our roadmap to the needs of the community. They need to speak.
I was just wondering, because you had all these... Oh, sorry, Irena.
Yeah. So I just wanted to add to this. We didn't discuss it during the session, but it's also a responsibility of our computational community to develop intuitive visualizations that would enable users to relate the results of the human cell atlas to perhaps the anatomical systems and the different organs and so on.
Go ahead with your question.
I was just wondering, because you've had some of these amazing jamborees, and have you been engaging with the biological networks to maybe set some of the questions that they are asking as topics for those?
So that's actually one of the plans, and the hope is that once there's at least some semi-functional version of the annotation engine to bring, again, at the level of grad students and postdocs where connections are truly made effectively, to bring a biological domain, for example, immune or something like that, and a bunch of computational scientists, and help them develop a common language through some type of annotation, analysis, and visualization tasks. That's on the plan, but we really want the annotation engine to be at the center of this, which means it needs to exist for that. Peter, do you want to add?
Yeah, I just wanted to add that I think in planning those jamborees, we do obviously work with HCA members, and including people who are on the biology side. And in the past, we've been able to actually get some data for the purposes of Jamboree. And obviously, if we're working with experimentalists, that would be leaning towards their own system. So hopefully in the next Jamboree and so on, we'll have more biological challenges as well.
Yeah, no, the community has been wonderful in helping us generate data just for the Jamboree, just to generate training data and ground truth data and some data that could be used for the Jamboree task. But I think the next level, as you suggest, would be to put graduate students and postdocs from bench biology and domain experts and some tissue atlas efforts, and sort of more mature computational scientists, mature in their computational skills, not in their career path. And I think that those ties are going to be what will really propel the entire HCA forward.
At the front, Tom, and then up the back after that.
So just related to that question, so I have a couple in our team back at home who were sort of watching the live stream, who have kind of like data science, database, programming background, who are now working with us on the biology. And I got a message like, oh, this sounds awesome. That was the actual wording during your talk earlier. And I was like, oh, how do I get involved? And I was like, oh, I don't know. So I said I would ask, and I'll ask now. So part one, and then part two, would you consider doing a Jamboree in Australia or somewhere down there? It's a fun place to visit. Just suggesting.
My current jet lag says that, here has been rough, but eventually we will have to get to Australia, but I think that right now the first task is to build these joint communities. Hopefully the HCA will also become big enough that there could be multiple local jamborees. They don't have to, I think that the Jamboree concept, once there's a lot of framework, because there's so much work to do, so many grad students and so many postdocs, that I'm hoping that, organizing these things are actually pretty time consuming. And they've, certainly drained a lot of energies from the AWG. So hopefully these will become scaffolds and skeletons to be replicated more locally and more broadly to enable more participation of more people. All you need is really 60 really engaged and enthusiastic graduate students and postdocs, and a bunch of more senior members with a clear view of clearly defined tasks, and a little bit of good food, some wine, and a good compute environment.
Right, Tom, so I'm taking that as a volunteer role for our Jamboree offsite. Thank you very much. And the next question at the back.
Well, I just wanted to add to this. So the other side, non-Jamboree side of getting engaged, so Tim Tickle talked about, I think, red box community, and that for data science folks in particular, I think that's the great avenue to get engaged. You could build your own portal, your own view on the data and so forth.
Cool, very, very interesting discussions that you had at the table. I want to get some clarification on the visualization question because it's something close to what I'm interested in. So I heard on the slides you had something about looking at single-cell ATAC-seq for like in genome browser style. So that's one thing which I envision as something more exploratory for hypothesis generation, et cetera. But I also heard about visualizations that are meant to communicate what HCA actually has done, which sounds a bit more, sounds different in terms of the goals for these visualizations. Can you clarify that? And for the second comment in particular about the other visualizations that were not on the slide. What I
so,
read.
I mean, right now, you want? Okay.
Yeah, I can take that. So that's something we didn't discuss on the table. But it's something that some of us have been thinking about, how we can relate the gene expression or the ATAC-seq expression, the ATAC-seq results or the spatial into the drawings of the organs and the anatomical systems and the different locations and so on. And that would enable users who have never really analyzed single-cell expression data or they have no knowledge of bioinformatics, or they're from medical background, to really engage with the data and ask their queries.
I see. So it's meant to be an entry point into HCA world, basically. Okay, thank you.
A quick comment or question.
When is the next Jamboree and how can we participate?
After I get a little bit of sleep, no, there's going to be a normalization workshop, which is not quite a Jamboree. And again, we really want to restart some of the Jamborees around some of the mixed, what we discussed, missing biologists and computational scientists. When will the annotation engine be useful? In a month, right?
No, just to clarify, this stuff is still on paper, so we're hoping we can build this quickly because a lot of the components are standard, but it's not. It doesn't
Yeah,
exist.
so I mean, I think realistically, since we don't want to put a stop to this, we'll aim for something in next late spring or summer that's more Jamboree style, because we do want to handle the normalization workshop, which is going to be slightly a different format and more, a bunch of more PIs brainstorming at this really hard nut and trying to find some avenues for solutions. I will say that organizing these things involve an incredible amount of resources, both financial but also times, and we really want to keep the Jamborees up to the standard that they've been at till now. But there will be more.
Will there be a public announcement for the normalization workshop or the Jamboree? How will we know about it in advance too? And how can we participate?
I think, again, one of the problems is that these things are fully funded by others. So the participation till now has been fully funded participation, so we couldn't do completely open access. We are thinking of ways, which have not yet been completely determined, into combining some selected slots of some people who have been engaged in the problem, and we want their input because they have some expertise, with some people who sign up, and thinking of a best way to fill up. There are only 60 slots. So we're thinking of more equitable ways to, one, fill up those 60 slots, and on the other hand, ensure that the right expertise so that the Jamboree, for all its effort, will be successful, are represented in those 60 slots. And we're still discussing how to do that.
We're going to have to take this offline. Sorry, Al, I know you keep putting your hand up. But it sounds like a few people are interested on how to get involved, that it does take a lot of work, but maybe there's thoughts around how we might scale this without the same people organizing it over and over again.
Yes, there's thoughts around that too. Anyone who wants to volunteer to organize rather than participate, also please send me an email.
And I reckon there are probably people in the audience who would. So I think that's a good point to finish on, and then we'll move to our next speaker. Thanks very much. So next is Beyond Single Cell RNA-seq from Will Greenleaf, and I know he'd like to clarify to you all whether it's ATAC-seq or ATAC-seq. Is that correct?
Yes, I've used that joke one too many times now. Everybody knows it. Okay. But I do say ATAC-seq. Okay? ATAC-seq. Okay, great. Uh-oh. Whoa, Beyond Single Cell RNA-seq. All right, so this is, we were in room C, and I have some wordy slides here. That's my fault. The only reason there's slides at all is because of my scribe. Where's my scribe? Stand up. Give her a hand. It was just me in there. She was the one taking notes. Thank you so much. So edited notes from there. So okay, we sort of broke this into two different sort of parts. One first starting with what brainstorming. What are the cool non-single cell RNA-seq methods we might imagine doing at some point to merge with the very large amount of single cell RNA and spatial transcriptomics type data. Okay, so what are these? Well, a lot of people said protein, surface markers, intracellular. A lot of people said chromatin, accessibility, ChIP-seq, higher order chromatin interactions potentially. Methylation was a big one. Cell-to-cell interactions, information about what cells talking to whom. And then there was a lot of other things that people were interested in, maybe because of the biology that they were interested in, including protein modifications like phosphorylation, the secretome, metabolites, mass spec, i.e. sort of an unbiased sort of measure of proteins such as it is, non-poly RNA, non-coding RNA, microRNAs. DNA itself, that's potentially interesting in a number of contexts, including tumors, mitochondria, lineage tracing. There was a lot of interest in potentially in sort of a developmental context. Who is touching who? That sounds racier than it meant to, but what cells are touching what cells, potentially with slicing. RNA modifications, which was partial credit if this is non-RNA, but I think it fits. It fits. EM tomography, Raman, Hi-C, and I think there was actually a lot of interest also in just straight up cell shape, morphology, and high content imaging, and also cell cycle. Okay, so I came up with these questions, so maybe I was a little guiding here, maybe. But I also wanted to probe the possibility of data types that might allow for phenotypic sorting or the probing of functional DNA. And I think there was a lot of response to this. I think there's a lot of interest in this idea of how we might use HCA or move in the future toward function, trying to understand cell function, both with surface markers, which can be measured. There's multiplex assays, site-seq and other of its ilk, and potentially also ways of using RNA and the large data sets that already exist for prospective identification of potential surface markers that are interesting, and then validation of those markers. Also, cell membrane shape morphology, people were interested even in patch clamping in terms of function. And I think the discussion got even broader, where people were talking about potentially going so far as to actually perturb regulatory elements or genes and then start to infer function in sort of a more perturbative way instead of observational. So there's this broad question of whether function, actual cellular function, is part of this annotation, and in some sense it would be a gold standard if we could get to the function. And what kinds of ways could might we add functional assays? In some sense, HCA, it was observed, is more observational, and eventually it might be interesting to think about ways to link just this observational mapping sort of work toward a functional output. So there was a lot of discussion about, I polled every room to ask how many people had actually generated non-RNA single-cell data, and I think in the end it was about 10% maybe of people. And so I was really interested in do people just not want to do this, or are there barriers to doing this that prevent people from doing it? And I think a lot of people actually would, given finite resources, put some of those researches on non-RNA methods, but there are a lot of potential barriers that prevent people, I think, from doing that, including, well, first of all, RNA-seq itself is very highly scalable, and a lot of these other methods are harder to scale, and it's potentially more expensive. And there's a whole raft of antibody-based method challenges in some sense. So there's this dichotomy between antibody-based versus non-antibody-based methods, and it's hard when you have antibody-based methods to do a discovery. You sort of have to already know what you're looking for. Also, there's a dichotomy between surface markers and intracellular staining. Intracellular staining gets you closer to the function, but surface markers are what you could use prospectively. And of course, this is all convolved with dissociation. Some markers might be lost, and of course, if you're working with nuclei, which is often what has to happen in complex tissue, there aren't any surface markers, so you're SOL, as they say. So also, antibodies are expensive. People are exploring non-antibody-based methods, and often antibodies are really tailored only for blood, where the majority of this sort of surface marker-based work is done. There's really no guarantee that antibodies will work, and you often have to test them individually, which becomes very expensive. And there was this idea that there might be a need for a repository of monoclonal antibodies, so that basically this object that you're using to carry out experiments won't get lost. But there was also some positives with respect to antibody DNA analysis. In some sense, it's often easier because it often builds on things you're already familiar with, such as facts and things like that. Okay, and we maybe covered this a little bit, but requirements for this targeted analysis, RNA, you shoot first and ask questions later, you get all of your genes in an unbiased, reasonably unbiased way, and other technologies, especially this kind of sort of antibody affinity-based markers, you really have to know what you're looking for, or at least select a subset that might be useful. And then there's a whole other class of barriers to sort of omic data other than RNA-seq. And frankly, I think there was a large consensus that much of this is in the data analysis and interpretation side. So for RNA, there's more tools, more papers, more documentation, more computational methods. And there's a need for more public data sets, and also more public data sets for non-RNA-seq data. But also it requires sort of irreducibly a big investment to build a computational team that's familiar with data sets other than RNA. And so companies or core facilities can often do part of this job, potentially for developing pipelines for alignment and maybe getting to a matrix format. But there's an irreducible challenge. At some level, you have to actually dig into the data and extract biological meaning, which requires a familiarity with what the data looks like, ways to analyze the data in coherent, rational ways. And I think that there was some interest in this potential for a detailed end-to-end tutorials that might start with raw data and go all the way through an analysis to figure in a text or a biological insight. I think there was a lot of interest in that. But again, this in some sense doesn't eliminate this irreducible need for analysis expertise that's distinct from RNA analysis. Also, there were some labs, commercialization is also a big sort of accelerant, I would say, with respect to non-RNA analysis. Some labs are comfortable with optimizing and doing things on their own, and other, maybe a larger set of labs are more comfortable with buying things off the shelf and things that have already been troubleshot, and you don't have to monkey with your buffers. And so I think there was a lot of weight, in some sense, for commercial kits to be broadly available. Okay, the other big topic that we tried to cover is multi-omics. So first, I just wanted to sort of get at what kind of biological insights that these sort of multi-omic data might enable, and there was a number of things, and this is sort of a list of them, including this idea of developmental biology and lineage tracing in transitional states, and trying to understand sort of Waddington landscape type analysis, and I think there's a lot of interest in 3' RNA chromatin and protein for this. Again, who is touching whom? These dirty, dirty people that were in room C. Does it, and then of course, does that affect a phenotype? Linkage chromatin state and RNA dynamics, and this was previously, I think, discussed by Donna, the idea of trying to get at correlation and causation, or ideally causation through correlation, I guess. And then there was a lot of discussion about what would be the highest return sort of combinations of data to acquire. And everyone always said RNA accessibility and protein, everyone wants everything all the time. There's a lot of interest, again, in cell-cell interactions. RNA, straight up DNA and RNA, especially for cancer applications. DNA and DNA, I'm not sure what that means, actually. It could be accessibility and methylation, or actual genomic information and accessibility. And protein and DNA, again, all the combinations you can think of they were interested in, but everyone, the first thing out of their lips was, oh, RNA, chromatin, something, and protein. That's what everyone wants. So yeah, okay. Okay, and then we talked a little bit about there's always going to be a trade-off, almost certainly, between getting multi-omics data in the same single cell and potentially getting the same data set in distinct cells that came from the same sample. And how much should we focus on this trade-off? What should we be thinking about this? And there was a lot of consensus that we really do. It would be very nice to have a ground truth where we get multi-omics data, even if it's slightly degraded on both fronts, for example, from the same single cell, because that especially helps us comparing bioinformatic methods for linking distinct data sets. Right, and so along the same lines, the idea of doing separate measurements and then having a few ground truth measurements that potentially link them. And then there was a lot of thought around this idea of aligning, for example, chromatin information in RNA. Timescales are often different with respect to the lifetime of RNA, whereas chromatin is whatever it is when you assay it. And this is a challenge both for distinct analysis and also the combined data sets, because if you're assaying a cell and you're getting a lot of cytoplasmic RNA, all that stuff happened hours ago, potentially. So, okay, we also generated a few questions, which maybe we didn't really have a lot of time to discuss or didn't have answers to, but given what's in the HCA, are there features or metadata that we're not collecting that might be important later when potentially we start generating data that is non-RNA based, or more data, I should say? And then how can we make collecting non-RNA-seq data in the future most compatible with the RNA-seq data sample collection already in HCA, so that we might have naturally multi-omic data from similar, either the same samples or similar samples, so that the data can be more synergistic when it's generated. Okay, that's what I got. If there's anyone from anywhere that remembers anything else that should come to the fore, or if there's questions, I'm very happy to take them. There we go.
Aviv.
That was awesome. On the last question about how do we make sure that the samples can then be used for something else, I think for at least many of the assays that are not RNA, you can do them with frozen samples.
Yep.
Either just with sections because they're not cellular samples, or at the nuclear level because they're kind of the DNA and the attack, and
That's
that
right.
kind of is anyway only in the nucleus.
Yeah.
And for them, really, the most important thing is preserving samples rather than using all of them up. And I think Joachim showed earlier for the developmental atlas, and I believe that's also true for many of the others, also in HubMap and other of the efforts, that there is more and more systematic sectioning going on, and I would strongly advocate for us to really follow that. That was also the way it was laid out in the white paper.
Sorry, systematic sectioning. That
Meaning
means
you section systematically across the sample, and you predefine which of these sections will go for certain uses and which sections are basically going to be preserved.
Yeah, that's great.
Yeah, or you split a sample when it's still fresh, and you freeze down part of it, and that part can get sectioned.
Absolutely.
Yeah.
I think
Dana
that's great.
wants to say.
So I definitely agree with frozen and preserved samples so that we can go back to the same sample because it's really valuable. But I do want to stress that as much as what we've seen today is awesome progress and an incredible amount of cells, we've just begun. This is just the very beginning. So I think we really should focus on what is the most informative, valuable data to collect in the future, and not change our schema too much based on the path if there's something much better for the future. This is really early steps, and we should really think about the best experimental design for the future when we can, particularly through preservation of samples, definitely backward compatibility, but not let us hold us back.
I'll just add that one of the reasons to preserve samples is not because of the data that was already collected from another portion, but just because these are human samples. Many of them are very precious. People consented to sample collection, so maximizing how we handle them, I think, is going to be important in a way that's different from other studies that we do in non-humans.
Anybody else?
I have a question.
Alrighty.
Flow cytometry has been around for ages.
Indeed.
And we're getting up to many, many markers. If we started the clock today and said, everyone upload whatever data you have, annotate it well, do all your single stain controls, would that be useful to the HCA, or is it in the too hard basket?
Yeah, I think that's a open question. I would imagine. I think that my intuition is that the majority of that data comes from blood, and I think but also tissue resident cells and things like that. So I am not as familiar with the challenges of normalization and in flow cytometry, but I think that there's many of the similar challenges that we are dealing with respect to the RNA-seq data. So I think that it's a non-negligible challenge to get that to happen. And then the question is is there a community that would use that in a serious way to sort of retrospectively, dig into different questions, and I'm not as sure of that as I am for these really highly multiplex molecular assays like RNA-seq, but I'm happy to be contradicted by anybody.
I get the intuition it's in the too hard basket, and that everyone would want to, do site-seq plus transcriptome and start kind of fresh, especially because you can add many proteins rather than just max 20 or something like that. But anyway. It's hypothetical. Any other comments or questions?
There's synergistic data sets, I think, obviously, too. So, all right.
No further comments? All right, let's thank William. Last but not least, spatial genomics, led by Joachim, Mike, and Ambrose. And we'll hear from them now.
Cool. Good. Okay, I know I'm last. I'll try to be quick. So we were doing the spatial genomics group with Mike and Joachim. Fair warning, I'm going to ask you to come up here and take questions with me, so be prepared. We started by polling the group's characteristics. We wanted to know how many people are actually working with spatial data right now. And it looks like only about 5 to 10% admitted to actively working with it, but we then polled them how many were interested in spatial.
approaches, and only 50% said yes. So I'm going to guess there's some kind of false negative rate going on there. We were trying to ask big questions around what are spatial approaches needed for in terms of constructing a cell atlas, and how much spatial data will be needed. Understandably, there was also lots of interest in the answer to the question, what assay should I set up in my lab, which I'll spoil. We don't have a good answer for. So in terms of key use cases enabled by the technologies, there was the obvious identification of rare cell types. When you look at sequencing data and you get something that's really rare, it's often difficult to distinguish between just an artifact that you've managed to pick up and a real cluster, whereas when you see that in an image, it's often a lot clearer that that's truly a real signal. And then the identification of the effects of those rare cells on the microenvironment. So what are those pairwise interactions and what can we learn from those? Also, the visualization of the data and using that to drive feature discovery and hypothesis generation. When you have a big volumetric image, you can play around and look for things you might not have expected a priori with your experiment. And then the next question was, what QC measures do we have? How can we trust this data right now? And so we walked through some things involving using code books that have blank codes for methods that have multiplex identification, the fraction of your gene signals that map to those blanks, it's the false positive rate. General questions that are similar to what you would see in single cell approaches, like the total number of molecules you see or the number of molecules per spatial area. The dynamic range of your probes, what's the minimum detected rate and the maximum detected rate for your cells. And then using multiple probes or antibodies against the same target. So describe experiments where you expect co-localization and you have a good gold standard. And finally, the use of biological priors. So define codebooks that have mutually exclusive markers or markers that you expect to co-localize. The next question we asked was, how much spatial data is needed to construct a draft of the atlas? And the answer was pretty conclusively not a lot, probably just enough to construct a scaffold that you can map all the rest of the data down to. But there was a good presence of mind in the audience to identify that while there have been good examples of that working, for example, the data set from Simone Codolupi and, give me the name. Sten, thank you. That was shown to map really nicely to RNA-seq with only like 16 to 32 markers. There was tons of effort that went into the identification of that probe set. And that information content's really important to enable that mapping. So I think it was left as an open question what the resolution of the scaffold and the common coordinate framework would need to be to enable those types of approaches. Another question that garnered a lot of interest was, do we need 3D data or is two-dimensional data adequate? Understandably, the general consensus was that more is more. But I mean, if you ask a child, Mike put this very nicely, if you ask a child if he wants 3 cakes or 2 cakes, you're going to get one answer all the time. But it's a lot harder to answer that question in a cost-neutral space where you might be offered 2 3-dimensional atlases of 2 patients versus 100 patient replicates in 2 dimensions. Kind of hard to say what you'd pick there. One suggestion, which seems reasonable, is to build a few 3-dimensional atlases and get a sense of what the variability is across that feature space. Do a bunch of 2-dimensional projections and just see if the information content's retained. Once you know that you can map down into 2 dimensions, then start building out a broad atlas. It was also a question that we couldn't come to a good answer to, whether there are specific tissues for which 3 dimensions is just not necessary. Perhaps you can just start from 2D. Another open question that we weren't able to answer is what's the right imaging resolution? Subcellular resolution has obvious benefits of being able to apply more biological priors, for example, apical basal in epithelial cells. But it's also clear that spatial transcriptomics, which doesn't even have single-cell resolution, is a very, very powerful approach to generate spatial knowledge. Finally, I was trying to ask some questions about how can we effectively share this data and how can we unlock the single-cell RNA-seq community to start working on these spatial approaches. And I'm involved in this base-TX benchmarking effort, which has 10 groups generating one data set each, and that's going to be 250 terabytes of data. It's quite a lot. If 10 of you wanted to download that from AWS, where we've put it, it would cost CZI $250,000. I'd hope more than 10 of you would want to work with the data, and so questions around data accessibility are really interesting to us. There's really only two options, which is like one, either you guys all set up cloud accounts and work with it, or we set up at the post office and hire 2 people to just mail hard drives full time. So just general questions around data availability feel worthwhile to address. And those were the main topics. I'm happy to answer questions with Joachim and Mike.
Some really fascinating challenges there, especially for spatial. Any comments or questions? Sham.
So data analysis is still a big challenge in this area, as you know, and you're actively involved in it. It would be great to have ground truth data sets for some basic data analysis processes, like say segmentation or something like that, and yeah, maybe other steps in the pipeline also. So maybe you could comment on what ground truth data sets are available right now.
Well, ground truth's a really interesting question. I think we should just start with getting data sets out. We are working towards that. At least for the space text data, we intend to put that all publicly available on an AWS bucket. There's 4 data sets there right now. If anyone wants to come bother me, I'm happy to share the link to that. It's just raw image data for some 2-dimensional, some 3-dimensional data sets. We have sequential SM fish, we've got MER fish and CT sequencing, and we'll very soon have a seq fish, and we should probably figure out how to put the spatial transcriptomics data up there as well.
Okay, that's a great start. So I guess we can email you and find out how to access it. Okay.
Yeah, you can email me. Anyone can get in touch with us on the HCA Slack or CZI Science. Both of those are open, and we'd love to have more participation in either of them.
Awesome, thanks.
Come on, space. It's the final frontier. There we go, at the front.
You mentioned a little bit about the resolution for the sampling, not how many markers to measure, but how many, say, fields of view or the tissue and so on. Was there any sense of how to even go about thinking about this question? Especially in the context, these organs are kind of big. And for imaging, some of these assays take, maybe Mike has a sense. He's also a trained pathologist.
Just one second.
You have to wait for a mic because of the live stream.
I think that probably just as important of a question as it's gonna be pretty dependent on an organ per organ basis. Like I think, for instance, GI, because of how long it is, you're probably going to have to take many more fields than you were in other types of organs. I don't think it necessarily even scales with the size of the organ either, because liver, for instance, you probably don't need as many as you do in sort of smaller organs that actually have a higher degree of complexity. And so I don't think the number is intractable, though. I think depending on the organ, maybe not excluding the brain and the nervous system, all of that, but everything else probably, somewhere in the order of, less than 100 fields or something.
For the whole organ?
I think so.
For the whole organ?
I think so. If you pick
my day.
I mean, this is going to be relying on
100 per individual.
I think 100. I think you have like 100 fields per. Like if you're talking about being able to like map to look at all the different. So I am thinking from the standpoint, if you're saying sort of. If you were to ask a pathologist, how many fields do you think I need to look at, say, in kidney to get a good sampling of, all the different histologic structures in sort of a normal tissue?
Yeah.
I think they'd say, 15 to 25 people, something like that.
50 to 25, but in terms of the anatomical diversity of the organ, you would need sections per region or?
Sections per region, yeah.
Okay, that's not terrible at all.
No, I don't think it's that. I mean, you could go more, but I think you're starting to rapidly hit a point of diminishing return. You know, I mean, it's now. I mean, depending on the complexity, the organ might be a lot higher, but I don't think it's crazy high. I think it's upper end is maybe a couple hundred or something like that.
So I think this is one of the questions that we'll need to grapple with the most in the near future. Because if it's actually more or less manageable matters a huge deal in terms of moving from just kind of a tissue level to an organ level, and that's something that we really need the pathology community in on. Yeah.
Just a comment on the fact that, I mean, you said that using 15 or 20 probe, you can probably identify the cells and put them in the right position. But I really hope that we can have a higher resolution, because very often RNAs are positioned in different parts of the cells, and you can infer very much about the function, and they have localizations that are very important. It seems that in a tissue is what we never had, and I really hope the people can be very ambitious and try to challenge beyond the assignment of cells to a kind of framework.
Yeah, I think there's a really interesting trade-off in terms of your ability to clearly detect a small number of transcripts versus get a lossy sampling of quite a large number. Single molecule FISH has been held as the gold standard to which RNA-seq was compared, right? And so if you've got 30 of those, you know with very good confidence you're getting the RNA transcripts. In contrast, a MERFISH or a seq-FISH approach, which has a very large codebook, may detect 1% or less of your transcripts. There's also a need for computational development, I think, in those spaces where you have very large codebooks, and we need to be able to estimate our confidence in those calls. So getting more computational biologists involved in that community and those questions could be very valuable.
Thomas?
I have a question from one of our grad students back in Sydney who just slacked me. Is there any work at the moment on linking data sets from multiple organs? And this kind of relates to, so some of the atlases are organ specific, so a liver atlas or something, but the immune atlas obviously has this kind of multi-compartment aspect to it where I don't really care what the liver atlas looks like, but I do care about I'm sorry, I didn't mean to say it like that. I mean, in the project, I care about the immune cells across different tissues. And so is there any work on linking data sets across tissues? If the answer is no, that's fine, but I'm curious.
I know Goji's done some interesting work on stromal cells across the tissues that he was looking at. I'm sure that there are more examples as well, but I'm not aware of them.
Just to answer that, I think that's now an emerging theme. I think it was a very difficult question to answer when there were no tissues, and it's all of a sudden very possible because there are a lot of tissues. For the spatial aspect, I think it's still reasonable to say let's let the tissue guide the spatial sampling, then we can compare. But from a marker point of view, it is interesting for assays that require you to choose markers. It is interesting to think about kind of a pan-tissue analysis from the cellular side in order to define pan-tissue markers so that there isn't too much specialization on the marker side. But my gut feeling is that either way you'll be fine, because even if the markers are different, the covariation will save you, as it does save us every single time in this field, and so we should be kind of okay. But it's a good question.
So we're going to round up soon. Before you leave, I just wanted to plug both the spatial and the Jamboree aspect. At Oz Single Cell, we are holding a Jamboree on spatial technologies. You're more than welcome to join, July 21st, and we're going to get some data from spatial transcriptomics and get a group of people to start working on it. So hopefully that's the start of many Jamborees to come. Let's just thank our final speaker, and I think there's a group photo now, is that right? All right, so round of applause and then listen for further instruction.